Hello everyone, my name is Anwar. Glad to see you on my Bioculture channel and welcome to my SAT biology course. If you haven't subscribed yet, then be sure to be subscribed now, press like and share this channel with your friends and colleagues. Today we'll consider the third type of macromolecule, nucleic acids. So before we get started, let's dwell on what nucleic acids are. Nucleic acids are polymers made from monomers called nucleotides. There are generally two types, namely DNA and RNA. Here we'll focus on the structure of these molecules. You might think, what should you know in this section? The first is what the nucleotides are made of. They consist of a phosphate group, sugar and a nitrogenous base. The second is that DNA is double-stranded and RNA is single-stranded. Also, in the polynucleotide, adjacent nucleotides are connected by a phosphodiester bond, which consists of a phosphate group that binds the sugars of two nucleotides. This bond leads to a repeating structure of sugar-phosphate units, called the sugar-phosphate backbone. Please note that nitrogenous bases are not part of the backbone. And fourth, the two free ends of the polymer are distinctly different from each other. One end has phosphate attached to fifth carbon and the other end has a hydroxyl group on third carbon. We call them 5' and 3' respectively. I think everything is clear here. Let's move on. You might have guessed that DNA and RNA differ at the nucleotide level. So, nitrogen bases are shown here. We distinguish between two groups, namely purines and pyrimidines. The first group consists of two rings, and the second of one respectively. Also, there is a certain order of nucleotide binding to each other. In DNA it looks like this: AT, U in RNA, G, C, where the number of bars indicates the number of bonds. Thus, both groups are present in the structures of DNA and RNA. However, they differ in only one nucleotide, thymine in DNA and uracil in RNA. So, the next nucleotide structure is sugar, namely pentose. In DNA it is deoxyribose and in RNA it is ribose. So, what are the differences? The differences are that there is oxygen on the second carbon in ribose, but not in deoxyribose. In summary, you have to understand the details. What are the differences between DNA and RNA? Let's refresh your memory. First, DNA is double-stranded and RNA is single-stranded. The second, thymine in DNA molecules and in RNA uracil instead. Third, sugar in DNA is deoxyribose and in RNA is ribose. Okay, on this our fifth lesson ended. If you have any questions, be sure to leave comments under this video and I will answer you. As usual, I will leave a link at the description to download today's slide. I hope it was the most useful for you. And if you haven't watched my previous videos, be sure to check them out. I will be waiting for you in the next lesson. Всем большой рахмет!